Welcome, everybody, to the latest episode of Star Cells in God. This is the podcast where we explore how the latest discoveries in science provide evidence for God's existence, God's nature and character, and the reliability of Scripture. This podcast is sponsored by Reasons to Believe. If you want to know more about Reasons to Believe, I invite you to visit our website, www.reasons.org. Also, you can follow us on social media, RTB_official. And then last but not least, go to our YouTube channel and subscribe, Reasons to Believe 1, and set the notification so that you're reminded the next time there is a new episode of Star Cells in God. My name is Fuz Rana. I'm a biochemist and a Christian apologist, and I'm joined today by Dr. Hugh Ross, who is an astronomer and also a Christian apologist and a pastor. And we're going to be talking about a couple of discoveries. I'm going to be talking about ATP as the cell's energy currency and how we can use insights about ATP to make a case for God's role in the origin and the design of life. And Hugh, you're going to talk to us about French fries.
French fries, and knowing that you're a biochemist, is I got to bring in some biochemistry. French fries and acrylamide. So I thought, hey, that's going to help. Fuzz is going to get excited about both topics.
Oh, yeah. Familiar with both.
Okay, that's great. Well, it's a group of Chinese scientists who decided to take advantage of the British bio data bank. Mm-hmm. Because in Britain, they've gotten a large number of their population that they've been following over the course of many years about their diet. And they're basically using that to try to develop some new strategies for improving the health, not only of people in Britain, but worldwide. And so what this Chinese science team did is they basically took advantage of the database. And they pulled out about 141,000 adults from the United Kingdom. And these were adults who were studied over a course of 11.3 years. Mm-hmm. But they only looked at adults that had detailed knowledge of their dietary practices over that period and also detailed knowledge of their mental health over that period. And so there were a lot that didn't make the cut, but they wound up with a huge database. 141,000 people is a lot of people. And so what they noticed in the database is that people in Britain who consume more than one fried food portion per day, so that'd be like fried eggs, a hamburger, French fries, anything fried. So if they consume more than one portion per day on average, what they notice is that subset of the 141,000 had a much higher level of symptoms of anxiety and depression. And we're talking clinical anxiety and depression. Because after all of us go through episodes of depression and anxiety, but clinical is where it becomes serious enough that you need to see a doctor, you need to get treatment, some kind of therapy. And so like those who are consuming more than one fried food portion per day, more than a 12% elevated risk of suffering from clinical anxiety and a near 8% risk of clinical depression. And so this motivated them to say, okay, well, what kinds of fried foods are really, and what they noticed was people that were eating fried potatoes, say more than, they had a serving of fried potatoes every day. And, a lot of people do that. They'll have a hamburger and fried French fries. That counts as 2 servings.
Right.
So, and as you can imagine, there are people that actually do that almost on a daily basis. But what they discovered was it's fried carbohydrates that especially give you an elevated risk. So fried eggs, a fried hamburger doesn't nearly drive up the anxiety and depression that if you, say, had fried carbohydrates. And so, and what they notice is the most common fried carbohydrate that people eat are fried potatoes.
Right.
Potato chips, French fries would be 2 examples.
Right.
But they said, other kinds of carbohydrates, especially the simple carbohydrates. So the study actually showed that having fried sweet potato fries, not nearly as problematic as white potatoes, because white potatoes is a simpler carbohydrate. And so they were able to discern that much. And then they said, well, let's dig a little deeper in this, because they cited research papers that noted that any carbohydrate, where they're cooked at high heat or they're fried, and so even if you're baking them, if you're baking them at high heat, you can cause a problem. But they're citing papers that basically said, carbohydrates that are either cooked through high heating or through frying produce acrylamide. And for a number of years, as you're well aware, acrylamide has been noted as something that's a neurotoxin.
Yep.
And they said, well, maybe that's the cause of the anxiety and the depression. So what they wanted to do is put that to the test. Now, it would be unethical to take a sample of humans and load them up with acrylamide and see what happens to them. So they said, we're not going to do that. So they needed to find an animal proxy. And they wanted an animal proxy where the animal is social, because that's kind of what's going on with depression and anxiety, is people have a loss of sociability. So the animal they chose was a zebrafish, because zebrafish, they're small, you can raise them in large numbers and study them, but they also have the characteristic that they're very social with one another. So they basically were feeding these zebrafish high doses of acrylamide and seeing what happens. And also they could sample those out and they check what's going on in the brain chemistry. So that's kind of what half the research is all about. And what they discovered is that these zebrafish, number one, they noted that when they were given acrylamide, that they would tend to isolate themselves from the rest of the zebrafish. And also they lost their desire to explore. They basically would just sit in one little area that they felt comfortable with. The other zebrafish would be interested in exploring. They wouldn't, and they weren't interested in socializing. And so they looked at that as clear symptoms of anxiety and depression. So they said that's probably explaining why people on a fried food diet are suffering higher levels of anxiety and depression. But also looking at the brains of these zebrafish that were being fed, modest doses of acrylic, what they noticed was that you resulted in elevated levels of GABA and glutamine in the brain. And again, there's research showing that it actually kills neurons and disrupts neural function. And so this explains, and then what really surprised them is they noted it disrupts cholesterol metabolism and glucose metabolism. So they're basically saying it's not only causing brain disorders and brain dysfunction, but it's actually causing a lot of other bodily functions. And it actually ties in with another dietary study, which basically says people who consume a lot of simple sugar, refined grains, and fried foods and processed foods, they have higher levels of cancer, higher levels of cardiovascular disease, as well as mental illness and several others. So they were saying, well, maybe the real culprit here is the fried foods. Sure, those other things are a problem. And so they're basically saying, is fried foods a more dangerous thing to consume than simple sugar, the refined grains, and the processed meats? And they said, yeah, it does come out. That seems to be the one that's really linked with disrupting cholesterol metabolism and glucose metabolism, which would explain why, okay, if you're tired all the time because you're not able to metabolize your cholesterol and your glucose, that could explain the depression and the anxiety. So, and the bottom line is they said, we need to do more studies because it'd be interesting to see if toast is a problem. If you're toasting white bread, that's a simple carbohydrate. You're bringing it to heat. And they hinted that it's probably the level of heat that's a concern. And so if you're lightly toasting your bread, it's probably not a big deal.
Right.
But they said more studies need to be needed. Basically, they were establishing, okay, we got enough here to realize consuming a lot of French fries or potato chips is not a good idea. But let's actually get some more detailed studies done.
Right.
And they were basically the first to do this kind of in-depth study. So they're basically saying, we need more studies like this. But having read the paper, and I got the paper right here, it's in the Proceedings of the National Academy of Sciences, in my opinion, they've done enough to warrant that, people should avoid
Right.
consuming more than one. And of course, what they want to know is, well, how much fried food consumption is a problem?
Right.
Having fried eggs 3 days a week may not be an issue.
Right.
But they certainly established
Right.
having a hamburger and French fries 6 days a week, that is a problem.
Right.
But it'd be good to actually get some numbers on this. But this is a health issue that's relatively easy to solve.
Right.
And their comment was more studies. And I think they even got a slide here of French fries. So
Now you're just rubbing it in.
I'm just rubbing it in. Well, I looked at this and said, isn't it interesting when you walk by a fast food restaurant, they always make sure that the smell of fried food wafts out. And if you're downwind from a fried food restaurant, you may have eaten 2 hours ago, but when that smell hits your nose, you feel hungry and you're tempted to go in. Maybe just avoid being downwind of a fast food restaurant would also be a good idea. So any comments? I mean, you're a biochemist.
Yeah.
You probably studied a lot on acrylamide.
Yeah, well, I have worked with acrylamide. Usually we would polymerize it to form polyacrylamide gels that we'd use to analyze, protein mixtures, or people would use it to separate, DNA molecules and things like that. So it's a very common chemical, and we were very, very careful when we handled acrylamide, for that reason. When you polymerize it, it's the polymer is not dangerous, but the monomer,
the monomer is dangerous.
Acrylamide is, and I'm just sitting here thinking it's interesting. That carbohydrates seem to be the culprit. I imagine that you have to introduce nitrogen somehow into the molecule. So probably carbohydrates reacting with amino acids or other nitrogen compounds that would be in foods naturally is probably where the acrylamide is being generated. So it's interesting to me in terms of how that acrylamide is generated, but I'm not surprised that it could be.
Yeah, that was missing from the paper. They didn't exactly show you how it's happening.
Right.
They just said, we know it is happening.
Right.
Because we can actually measure acrylamide production as a result of people eating these fried foods. And then with the fish, they could tell that something's going on there as well.
Yeah.
But it'd be really interesting to figure, okay, what is happening when you consume fried foods? Because maybe they can come up with a supplement that you could eat so that you could enjoy your French fries and not have the acrylamide problem. That
is remarkable in terms of just how interdisciplinary the work was, in terms of epidemiological data and then just some good old-fashioned food chemistry and some interesting, toxicity experiments.
And the fact that this British database even exists.
Yes.
So
yeah.
That's helpful.
Well, that brings up a concern that a lot of people have, is that those databases people are concerned about in terms of losing privacy, right? But on the other hand, those databases are incredibly invaluable. I think in Iceland, I think it's Iceland, where they have a relatively small population, and they've got similar kind of a database that covers the entire population.
I think it's even a genetic database. If I'm
not, yeah.
So it's 300,000 people where you've got a gene record of the entire population.
Yeah.
And basically what these Chinese scientists were saying is, it'd be good if we had more than just this one database.
Right.
So
yeah.
I don't know. So Fuz, are you going to stop eating French fries?
Well, I just won't eat them 6 days a week.
Okay, there you go.
All right. All right. Well, you are done.
I'm done. Sure.
Okay. Okay. All right. Well, we'll move on to my topic, and Hugh, cryptocurrency is in the news quite a bit these days. There's a lot of buzz around cryptocurrency because there are people that are claiming that by decentralizing currency, by creating the means for anonymous transactions, it's going to be disruptive in terms of the world's economies. And
Makes it easier to launder money, though.
it does, right. There are good and bad things with cryptocurrency. Well, it's interesting because people are also interested in it because they are investing in cryptocurrency. In fact, one of my daughters does cryptocurrency investing, and she's trying to get me to do it. And as I've read a little bit about cryptocurrency, I don't know that I fully understand what actually gives cryptocurrency its value, right? And I'm familiar with blockchains and all that fun stuff, and how Bitcoin is a new Bitcoin is generated every time certain blockchains come together, but I just don't understand it. So I'm not willing to invest money in something I don't understand. But this whole idea of cryptocurrency to me is fascinating because it really raises questions about what actually is currency, right? And I remember taking economic classes in college, which I enjoyed thoroughly. If I didn't become a biochemist, I very well could have become an economist. I enjoyed economics that much. But we talked about currency, in what actually currency is and how currency functions in economies. And, I'm of the opinion that that when it comes to human inventions, currency may actually be one of the most ingenious inventions human beings have ever produced, and that it may be more responsible for the ability of human beings to advance civilization than maybe even the invention of the wheel. Now, I don't know if you would agree with that or not, but currency is absolutely indispensable to allow economies to function efficiently. If you didn't have currency, we would be forced to barter, and that's one of the most inefficient ways for an economy to operate, and it would stifle economic growth. And I'm going to just tell a little story here to drive home that point, and that actually relates to the discovery I want to talk about. But in a sense, currency is basically any kind of medium that holds economic value, right? And if you didn't have currency, let's say that I worked for a farmer who raises citrus. And so after a day's worth of labor, he pays me by giving me citrus fruit, right? Grapefruits and oranges and lemons. Now I need a pair of shoes. So I go to the cobbler, and the cobbler is willing to sell me shoes, but he doesn't want citrus. So I got to find out what he wants. Then I got to go to that person that makes that and hope that that person wants citrus fruit. If he doesn't, then I've got to go to somebody else and find out what this guy wants and go to that person. And so you wind up with this.
You got to do all that before the citrus goes moldy, right?
Yes, right. Right. So in other words, it's an incredibly inefficient system. And if I finally find somebody who's willing to take the citrus, then I've got to work my way backward in order to get a pair of shoes. Whereas if you have currency, which is just simply any medium that, again, holds economic value, then I can receive currency for my day's labor, and I can use that currency to purchase whatever I want, with that vendor being confident that by receiving that currency, they can use it to make a purchase that they want. So the idea here is that currency is really this ingenious human invention that makes highly efficient economies possible. Now, it's interesting that the cell makes use of currency as well. And this currency in the cell is in the form of a molecule that stores energy. So instead of having economic value, what's important in the cell is energy.
Right.
That energy is used to drive metabolic reactions. And metabolism is just simply a broad term that refers to any chemical process taking place inside the cell. But these metabolic reactions are producing building block materials that are then in turn assembled into the components of the cell. All of these reactions require energy. There are other metabolic reactions that are just simply necessary to maintain the cell in a living state. Other metabolic processes are necessary for cell growth, for cell replication. So metabolism, again, requires energy, and it's responsible for life being able to exist. Now, that energy comes from breaking down what we might call foodstuff molecules, carbohydrates, which we just talked about, fats, for example. And when they're broken down, the chemical bonds that are broken release energy that the cell then makes use of to drive metabolic reactions. Now, if the cell operated analogous to a barter economy, then what you'd have to do is couple one metabolic reaction to another metabolic reaction very specifically. And if that particular compound isn't available in the cell, then the necessary metabolic reaction can't take place. So it would be such a highly inefficient way for metabolism to work in a bartering type of manner that I even question if life would be possible. But what the cell does is, again, ingenious. It makes use of a compound called ATP as the cell's energy currency. So when fats or sugars are broken down, that energy that's liberated is ultimately used to produce ATP, and then ATP can be broken down to liberate energy to drive metabolic reactions. And it's universal in the cell in terms of its ability to function as this energy currency, and it's actually universal among all life forms. Every life form uses ATP as the energy currency, and so this is a cartoon showing the structure of ATP, and it stands for adenosine triphosphate. So it starts with a sugar called a ribose, and then attached to the 1' position is a nucleobase called adenine, and then attached to the 5' position of the sugar is a phosphate group that's joined via an ester linkage. That's called the alpha phosphate. And then there's the beta and the gamma phosphates that are joined by a special kind of bond called a phosphoanhydride linkage, which is a high energy bond. And so when that bond is broken, it'll liberate energy. And in order to form ATP, it forms from the reaction of ADP, adenosine diphosphate, and an inorganic phosphate. And so this is showing the reaction in where the phosphoanhydride linkage is broken and energy is released for the cell to use. And you have this ATP-ADP cycle that takes place where when foodstuff is broken down, ATP molecules are formed from ADP and inorganic phosphates. And then when when energy is needed, the ATP is broken down into ADP and, again, inorganic phosphate. So you've got this cycle that takes place. And the ratio of ATP to ADP actually gives you information about the energy status of the cell. And so if the energy status is low, ADP dominates over ATP. If the energy status is high, ATP dominates over ADP. And so this is an incredibly elegant system that exists inside the cell that is really identical to the type of currency that we would use in economic systems. And it makes very efficient metabolism possible inside the cell. It's an ingenious system. And in fact, you could probably, on this basis alone, make an argument for God's existence and role in the origin and the design of life, kind of a watchmaker type of argument, right? That currency is a human invention, that it's an ingenious invention that makes efficient economies possible. And what we see is the direct analog to that inside the cell, where I see, again, the use of ATP as an energy currency as a brilliant, ingenious aspect of metabolic processes.
Now, this paper, did it discuss the trading of ATP that goes on inside the cell?
Well, we haven't gotten to the paper yet.
Oh, okay.
Yeah, but yeah, go ahead with that.
No, I'm just asking that question because to me, we've known about ATP for a long time, but I think what would be fascinating, how do the different components of the cell actually trade ATP? How do they make the currency work?
Yeah. Well, I mean, the ATP molecules are everywhere.
Right.
And most of the ATP in eukaryotic cells is made inside mitochondria, and there's actually an ATP transporter that will move a molecule of ATP.
So that's the bank inside the cell.
Yes, exactly.
Yeah.
Or the mint where the money is being printed.
Right.
And then the ADP that's been spent, or that's been generated from ATP that's been spent, gets pumped into so it's an anti-porter where ATP goes out, ADP comes in, right? And then inside the mitochondria, more ATP is generated. So you've got these different pathways that are moving the ATP in and out of mitochondria, but it's essentially available in solution everywhere in the cell. So the components have ready access to it. But it's interesting because ATP again is universal, which means that it must have been present as the energy currency in the last universal common ancestor. If you're thinking about the origin of life,
yeah.
In evolutionary terms. In fact, it probably would have had to have been present in organisms that preceded LUCA. And so it's a question, really, where does ATP
come from originally?
Yeah, how does it originate? Because you cannot, at least I struggle as a biochemist to envision a scenario where you have a bartering kind of metabolism, because that would be so inefficient that it would be very difficult for the protocells to become more and more complex. You almost have to institute the ATP as the energy currency all at once, because it's such an integrated system with respect to metabolism. And it's not just simply that the cells make use of ATP. That ATP has to literally bind to either the active site or allosteric sites on proteins. There has to be this high affinity and this ability then to either break down that bond, the phosphoanhydride linkage to produce inorganic phosphate, or transfer the phosphate to, an OH group to make a phosphoester. If you can't do that, then, the energy currency is going to be useless. So it's not just simply having ATP available, but you literally have to have the machinery in place for each enzyme in the cell to process ATP. So it's such an integrated system that if it doesn't come into existence all at once, you're not going to get there in a stepwise manner, because you might be able to envision very simple protocells where there's a bartering kind of economy going on, but you can't get much more complex than that. Once you start growing the complexity, the inefficiency would make really any kind of evolutionary progress beyond that very difficult. So it's a very powerful argument, in terms of really, can you explain this origin through chemical evolution and the similarity to human economies, human inventions that I think you could use to make a design argument, a type of watchmaker argument. But now the big question on the table is this: why ATP? So biochemists are interested why ATP was selected as the energy currency. So one model would be, this is just a happy accident of evolution. It's just historical contingency. The other explanation is maybe there actually is a reason why ATP is chosen. And, over the years, biochemists have suggested different ideas, but there does seem to be a rationale for ATP. And as I pointed out, when we looked at the structure of ATP, it's essentially not only functioning as an energy currency, it also doubles in its function as one of the building blocks to make RNA molecules. And so there are 4 nucleotides that are used to make RNA. It would be ATP, GTP,
right,
CTP, and then UTP. And so it's interesting that ATP doubles as an energy currency, as a molecule, as well as, again, one of the building blocks of RNA, because what it does is it now links the energy status of the cell to gene expression. So the information that's in DNA codes for proteins, and you don't want the cell to start producing proteins if the energy status is low. If the energy status is low, not only will you not have enough ATP, but you won't have enough of the building block materials to make RNA. So that linkage between energy currency and gene expression, to me, is probably why ATP is the energy currency. It's a brilliant.
Now, is this an issue for DNA or not?
Well, it's not an issue for DNA because DNA is using ATP, but it's a deoxy, so it's a different sugar.
Different sugar,
right?
Okay.
So and actually using ATP versus the deoxy version of it, which would be the building blocks for DNA, makes sense because it's actually more energetically expensive to make the deoxy ATP versus the ATP based on ribose. Removing that OH group and replacing it with a hydrogen is an energy expensive step. And so you're using ATP as not only, again, because it's part of this pool that links gene expression to energy status, but it's relatively inexpensive to make. In fact, if you compare ATP to GTP, it's less expensive to make ATP for the cell than GTP.
Wow.
So that makes sense, right? Because it's because the nucleobase is more complex in GTP. Now, why not CTP or UTP? Well, it turns out having that purine, which is a fused two-membered ring versus a single ring, which you see in UTP and CTP, again makes sense because the purine is more hydrophobic and therefore is going to interact more tightly with proteins, which is necessary for ATP to then be functioning as an energy currency. So in other words, there's a very elegant reason or set of reasons why ATP seems to be selected as the energy currency. It's not a haphazard outworking of evolution.
It'd be very difficult to come up with an alternate currency.
Right. It's hard to envision what would be an alternative. Right. Now, this leads us to the paper the long introduction there, slow burn. But this is a paper published recently by a team from the University College of London. And the lead researcher, the principal investigator, is a guy named Nick Lane. And he's becoming a pretty well-known origin of life researcher who is very interested in questions about the evolutionary origin of bioenergetics. So he takes a bioenergetics approach to the origin of life. And his question was, why ATP? And he thought, well, maybe there might be a prebiotic explanation for why ATP. And he and his team were speculating that maybe ATP was selected because a compound called acetylphosphate will preferentially phosphorylate ADP as opposed to GTP, CTP, or UTP, right? So, and this is a structure of acetylphosphate. It's a simple molecule, actually. And it turns out that people believe that this would have been possibly prevalent at high levels on the early Earth. And the reason for that is because at hydrothermal vents, you can get thioacetate, which is a high energy molecule. And that thioacetate in the presence of phosphate will form acetylphosphate. So it's a very simple reaction. And then it turns out that acetylphosphate is a very effective phosphorylating agent. It'll phosphorylate ribose, it'll phosphorylate ADP into ATP to form ATP. And so they speculate, well, maybe there's something about that reaction that led to the selection of ATP as the energy currency. So they did a series of experiments where they used all kinds of different phosphorylating agents, and they looked at those agents' ability to phosphorylate ADP, GDP, CDP, and UDP. And they discovered that acetylphosphate will selectively phosphorylate ADP to a much greater degree than the other nucleobases, which is very interesting. But the reaction requires iron in the +3 state. Any other metal isn't going to work. And they actually developed a mechanism for what they think is going on there. But the bottom line is they're arguing that this is a really unusual chemical reaction. that takes place that preferentially will produce ATP from ADP, right? And so these are some quotes that I pulled from the paper. The emergence of ATP as a universal energy currency of the cell was not a result of a frozen accident, but arose from the unique interactions of ADP and ACP. This implies that ATP could have become the universal energy currency of life, not as the endpoint of genetic selection or as a frozen accident, but for fundamental chemical reasons. Our results suggest that ATP became established as a universal energy currency in a prebiotic monomeric world on the basis of its unusual chemistry in water. It was very surprising to discover the reaction is so selective in the metal ion, the phosphate donor, and the substrate with molecules that life still uses. So let's kind of put this all together. What we have here is this very ingenious use of an energy currency that makes life possible, that allows us to make kind of a watchmaker argument for God's role in the origin in the design of life. It's very difficult to envision how you would get the level of integration you need for ATP to function as that energy currency through chemical evolutionary scenarios. ATP has this highly unusual set of properties that makes it ideally suited as an energy currency, which suggests it's not haphazard. There's a rationale there. And what these scientists have shown is that if you're looking at the origin of life from evolutionary terms, it's not natural selection that chose ATP. It's not a frozen accident, but it's fundamentally the chemistry, the laws of chemistry that allow for this highly unusual reaction that just happens to produce ATP as produce the ATP molecule. Now, there are some fundamental problems with this study. For example, the source of phosphate on the early Earth probably wasn't readily available.
Yeah, phosphate's relatively rare.
Right, and it's insoluble.
Yes. And what about the iron 3?
Yeah, iron 3 would be very rare. But most of the iron would be in the +2 state. So you could question the prebiotic relevance of the work that they did, and that would be legitimate to do. And, it's very tempting to do that. However, what is interesting to me is even if you adopt an evolutionary perspective for life, isn't it remarkable the coincidences that you see here? That the chemistry is such that it uniquely forms ATP. So if ATP emerged very early, now of course even generating ADP in a prebiotic scenario is highly unlikely, but let's just say for the sake of argument, you had ADP around, you had phosphate around, and iron 3 around. It's remarkable that the chemistry and the conditions of the Earth would be such that ATP would naturally form, and this happens to be that molecule with the just right set of properties that make it so ideal as an energy currency. And the whole concept of a biochemical energy currency is incredibly ingenious. So when you start stacking this together, you've got this really powerful argument for an anthropic principle, right? That the energy metabolism in the cell seems to be predetermined, prescribed.
You have to fine-tune the periodic table, and you got to fine-tune the chemistry for this to even be conceivable.
Yes, yeah. So the amount of fine-tuning, and this is just another example of fine-tuning in a prebiotic context, but it highlights the fact that if you are approaching the origin of life strictly in evolutionary terms, you've got a problem here because it's this unusual chemistry that's prescribed by the laws of nature that is actually responsible for biochemistry. It's not a frozen accident. It's not natural selection. And once you start describing life in those terms, it's deeply teleological. You can't escape design. You can't escape intentionality. You can't escape purpose.
Well, especially when you consider just how much ATP you need.
Oh, yeah.
Having a couple of molecules isn't going to do it.
No.
You need a lot of it in a concentrated region. And I look at the iron 3 problem, the phosphorus problem.
Yep.
It's like getting enough ATP for this to be realistic
Yeah. Yeah, I would agree with that. But again, you've got this, that even if you approach, the origin of life in evolutionary terms, you can't escape the design. I mean, you and I would say that design, is evident even in the the special properties of ATP.
Well, you need some kind of supernatural mind that's able to pick out the few iron 3
Right.
atoms that you need, get the phosphorus in the right chemical state, and assemble it all so
Right.
that you got enough.
Yeah, yeah. So anyway, I just found this to be, a fascinating discovery. A bit, convoluted to get there, but
Well, it reminds me of Fred Hoyle saying, trying to make the evolutionary model work is equivalent to speculating that a tornado goes through an iron mine, an aluminum mine, and out comes a fully functional aircraft.
yeah,
So,
Yeah.
got a similar problem.
very similar problem.
Yeah.
Okay, well, that's all I've got. I guess we can bring everything to a close here.
Sure. Okay.
All right. Well, thank you for joining us for Star Cells and God. I would encourage you to make sure you go to our YouTube channel, Reasons to Believe 1, and subscribe, and then also set the notification button so you're alerted when the next episode of Star Cells and God drops. Also, don't forget to follow us on social media, RTB_official. And then last but not least, make sure you go to our website, reasons.org. Until next time, remember, the more we discover about science, the more we have reasons to believe.